Hello students, so welcome again. So today we are going to discuss about the topic that is a DNA replication. So basically, the replication, it is a copying or sequencing of parent DNA strand into the daughter strand. Okay, so in molecular biology, DNA replication is the biological process of producing two identical replicates of DNA from one original DNA molecules. Okay, so this DNA replication occurs in all living organisms, so which is acting as the most essential part for the biological inheritance ones we can see. Okay, so next, what are the types of replications? So basically there are three types we can say, that is semi-conservative model, then the conservative, and then the last is the dispersive model. So this semi-conservative, so all in this what happened was all the atoms of one strand of this parent molecules are transferred intact and without rearrangement to one strands of the we can say progeny DNA molecules. So the other strand is formed entirely of a new atoms ones. So next, that is the conservative. When we say this, so in this what happened, the atom of the two parental DNA strands serves as the template of the two new progeny strands. So the new we can say parental strands remain intact, which is without rearrangement, okay, and remain together for following replication. So we can say there are two progenic strands are formed. So as compared to with when we see according to the picture in the dispersive, so in this what happened, all the atoms of each strand of the parents molecules appear in the we can say progeny DNA, but they appear as the large sections scattered throughout the length of both strands of the progeny, we can say DNA only. Okay, so it is the parental DNA, then the first replication and the second replication form. Okay, so these are the three types of DNA replications. Now according to this we can say there is a scientist, that is a Melson and Stack. He has conducted one experiment. So he decided the best way to tag the parent DNA, which would be to change one of the atoms in the parent DNA molecules. So as we all know, that nitrogen is found in the nitrogen bases of each nucleotide. Okay, so what they have decided, the scientists have decided to use a heavy isotopes of nitrogen, that is N15(1), okay, to distinguish between the parent and newly copied DNA. So the isotope of nitrogen had an extra neutron, we can say, in the nucleus, which made it heavier one. As we all know that according to periodic table, the most nitrogen atoms have an atomic weight of 14 only. Okay, so the atom is known as N14(1), but an isotope with an extra neutron, okay, neutron has a weight of 15. So what's added, so we call it as N15(1). So these scientists decided to start with the parent DNA molecules that only contain N15. So when we say if only N14 nucleotides were available during the DNA replication, so they could be able to tell which parts had come from the original double stranded and which part had been created during the replication process. So in order to make the DNA go through many rounds of this replication, so this scientist, that is Melson and Stack, they formed the reproduced powers of the common bacteria E. coli. So they made sure that the first batch of the bacteria contained only N15 DNA. Then they put the bacteria into a medium, okay, that only contain N14(1) atoms. So that way we can say whenever the bacteria reproduces, they would be forced to we can say incorporate the N14 into the new DNA. Okay, so the scientists they form the relax, they let the bacteria go to the work only. Okay, they form a multiplication of it. So with each new generation of bacteria, this scientist Melson and Stack took a sample, okay, during that period, so that they could see how the N15 DNA was being distributed in the daughter molecules. So we can say nowadays we can say many of the must be wondering that how could they tell us the difference between the N15 and the N14 DNA. So it is not like when we say actually see in the isotope of nitrogen. So again we can say, how did the scientists know that how much N15 was inside the each molecules? So so this answer is the atomic weight. Okay, the basic thing. So because N15 has one extra neutron, so it is slightly heavier than N14 one. Okay, and therefore it makes the DNA molecules denser one. So we can separate DNA molecule based on the difference in their we can say densities. So again we can say it we use they use as a centrifuge, a device that spins the test tubes and with a very high speed. When a test tube is spin inside the centrifuge, the content were pushed towards the bottom one. Okay, and the substrate that are heaviest sink further the down level to the tube, and the lighter substance afloat on the above the tube. Okay, so when we apply a centrifuge force to a mixture of the two types of DNA, the heavier N15 DNA, which was found to the lower level, and the N14 molecules was found in the upper levels, we can say. So we can say in every time this both scientists wanted to take a sample of the bacterial DNA, so they had to chop again we can say it is a method, we can say chop the microorganisms and empty all the contents into the test tubes. So they mixed in a salt solution, and then again centrifuge the test tube for many hours to make the substance separate out. So we can say then they used special techniques to see how far the DNA molecules hang inside the tube. So when they sampled their first group of bacteria, so this both scientists saw a darkened band in the test tube where the N15 DNA had sunk and gathered in the spot. Okay, in one spot, but after they let the bacteria reproduce it, they got much different results in their samples. So the DNA still sunk down in the tube, but not nearly as far as the first, we can say first generation. It was lighter form of DNA, meaning that it was not completely made with the N15 isotope after one replication. So the entire DNA had been converted to the hybrid of N15 and N14 DNA. So the DNA, we can say must be replicated so that the information it holds can be maintained. Okay, there's the main aim, we can say, and this can be maintained and passed to the future cell generations, even as that information is accessed or to guide the manufacture of the proteins. Okay, so these were the main content of this experiment, Nelson Stack experiments. Okay, so thank you very much, but do not spit in the public place due to this corona. Okay, so it is a very much risky. Even you avoid others, tell others to do not spit on the public place. Okay, thank you very much.